So we'll go ahead and get started. This is chapter 7. It's over the human body. This is going to be a several series section of the book because it's so important and it's so difficult, and there's a lot of information that typically is taught over several semesters in college that we're going to cover in a day. So there's a lot of information, so it is overwhelming. I understand that. Let me say this though. We're gonna break it into bite-sized pieces. I've said that, but there is no value in me telling you a bunch of stuff you don't need to know. Okay, in medical school, I know a lot of stuff that you absolutely don't need to know. Really, as a training physician, it's stuff that I don't need to know, but it's stuff that I've been taught. So that's not my goal. My goal here today is to teach you stuff that you're going to use and that you could see on exams and stuff like that. So know that if it's coming out of my mouth, it's probably pretty high yield and you probably need to know it. Albeit, this is overwhelming. It is a lot of information, so that's why we're trying to break it up into smaller chunks. So we'll go ahead and get started. So occasionally the book will ask, what's the difference between anatomy and physiology and pathophysiology? So I'll just explain that because it's easy. Anatomy, you can point at it, okay? So anatomy, I could just point at a poster and be like, that's the brainstem, that's the ureter, whatever I'm pointing to. You can point at anatomy, okay? And really when you can point with your finger, that's called gross anatomy. So gross anatomy is something that you can see with the naked eye, okay? Easy enough. And then we have microscopic anatomy, right? So you have to use a microscope to see it. So we might cut off a piece of the brain and want to look at it at the individual cells under a microscope, and that's fine. That's microscopic anatomy. The other word for this, occasionally used, just so you know, it's called histology. Okay, so that's like the study of small anatomy. Okay, microanatomy. In physiology, I told my friend the other day, anatomy you can point to it. Physiology, you have to use your words because it's explanation. Physiology is how does the body do what it does, right? So I can point to a femur, I can point to a knee, but to say, well, how does the knee work as a sliding joint, right? You would have to use your words to explain that. And you can't point to that. So that's a distinction, okay? And you'll hear me use a word a lot called pathology, right? Pathology is when something is wrong with the body. It's abnormal. Okay, that could be low oxygen, hypertension, high blood pressure, or cancer, or whatever it is. That's pathology. So pathology literally means something is wrong. Okay, and sometimes we use a word called pathophysiology. And what pathophysiology means is it's how does patho work, right? So you could point to a cancerous mass and say, well, that's cancer, right? Yeah, that's pointing, right? Pathology. But if I want to explain, the invasiveness of cancer, that would be pathophysiology, because I'd have to explain that. I'd have to use my words. So think physiology, you have to use your words. For the most part, anatomy, you can point to structures. And that's the distinction between the two. Now, you're going to look at a slide and be like, oh my God, this is ninth grade biology again. No one panic. It's okay. I've read your book. What's in your book is really about all you need to know. Okay? There's entire doctorate degrees dedicated to cellular structures. So we're not doing that. What's in the book is fair. It's what you probably need to know. I'm not going to read the book to you. I don't do that. But I'll explain what some of these things are, okay? So obviously our body's made up of trillions upon trillions of cells, okay? Individual cells, you've seen these before, probably under a microscope, as are plants and funguses and bacteria, right? They're just cells. And they vary. And here's the thing that I don't think students understand. This is an animal cell, right? This is a human cell. What's called a human cell. This is what's called a model cell, right? So your cells don't actually look like that, right? This is like a model. Like this is like the perfect cell of one of everything, right? There's one nucleus, there's a reasonably sized Golgi, and rough endoplasmic reticulum, and there's some centriole in there, and there's some vacuoles, and there's one mitochondria. In real world, that doesn't actually happen that way. Like, for example, like sperm, right? The little flagella, the thing that swims, has a lot of mitochondria back there because it needs a lot of energy. So there might be 20, 30 mitochondria back there, but there's not going to be a lot of, lysosomes, right? There's not going to be a lot of plasma membrane, right? It doesn't need to. So just realize this is like the perfect cell. And what actually happens in the heart versus the lung versus the chest or the skin or whatever. The cells look different in real world, okay? But students get lost on that. Like, this is a model. This is the perfect one with one of everything. Okay. Some cells have more, some cells have less. So that, obviously every cell, whether you think they're useful or not, has an individual function, right? Whether it's your hair or your eyes or your tongue or your nose or whatever, they all have different functions, and that function is determined by DNA. Okay. And uh, that's what they do, right? And in cells, as a generality, have to live. Okay, so they have to get messages and they have to get nutrients and they have to be told when to do their job, not necessarily to do their job, but when to do their job, because they're programmed to know how to do their job, but they need to know when to do their job, right? So, there's all these things that happen to cells. I'm not going to go on about this forever, so I'll wrap that part up. But um, it's worth knowing, yeah, cells do communicate electrochemically. Sure, that's important, right? So cells communicate through chemical messengers, right? Hormones, enzymes, blah, 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 right? Medicines, right? A lot of hormones and chemicals. But they also communicate through electrical impulses and electrical signals, okay? That shouldn't be surprising. But there's a lot of different ways that cells can communicate. And depending on what the cell is, it will depend how they communicate and the way that they carry out their jobs, okay? So this is just a general cell structure out of your book. And I could have put a different picture up here, but I didn't because it's a pretty good picture. So I'll just tell you some things in here that I think are behooved of you to know, okay, right? So let's just start with the nucleus, okay? So the nucleus is within the cell, and typically they want you to think they're centrally located. Not always, it depends on the cell. But it's like a little sac that has the DNA in it, okay? And within that sac, there's DNA machinery that can make RNA or start the process of making RNA and some other stuff, okay? But for the most part, I want you to know that the nucleus contains DNA. What is DNA? Deoxyribonucleic acid, right? Ribonucleic acid. It is our genetic code, right? So just like a code in a computer is a bunch of zeros and ones, in us, a code is made up of DNA of different chemicals, essentially proteins. And so that tells, it's a very, very long code, tells each cell what to do. Okay. Interestingly, the same DNA in my hair follicles, the same DNA that's in my heart. It's just they're expressed in different ways. So that's kind of an interesting thing. Your DNA is your DNA. Your DNA is unique to you. It's not, no one should have the same DNA as you. The only exception to that is an identical twin. I actually have an identical twin. But as we've aged, our DNA's changed through mutation. So really our DNA is probably not the exact same anymore. But at one point when we were first born, it probably was. So your DNA is unique to you, and and that's what makes humans unique, right? It's interesting. There's 7 billion of us, and we all look differently and sound differently and think differently and are different. Okay, and that's thanks to our DNA and along with some environmental influences, but really it's our DNA. Back to the nucleus. Okay, there's this thing called the nucleolus, right? So it's kind of the nucleus within the nucleus that really has a lot of tightly packed DNA in it. Okay, and then this thing right here called chromatin. Chromatin's loosely bound DNA that's actually doing its job, okay? Again, the same DNA in my hair is the same DNA in my heart. So when I'm making hair cells, you don't need the heart cells expressed, right? So what happens is chromatin, it kind of unwinds itself and says, this is the area that actually needs to do some work. We'll wrap everything else up real tight and put it in the nucleolus because we don't need it. So like for that particular cell. So that's what chromatin is. It's kind of unwound DNA that can actually be expressed and make proteins or whatever its job is, okay? So that's the nucleus. Got to know that, okay? It contains DNA. Moving forward, I'm just going to go in, I guess, clockwise motion because none of this will really matter. Now, ribosomes are little chunks of protein, more or less. You could think of them that way. Yeah, we'll just think of them that. Ribosomes are more or less protein within the cell, okay, or protein messengers. Glycogen, this is glucose that's on a really long chain. So it's kind of like energy storage, right? So we put a bunch of glucose and load them up on these long branch chains. And when your cell needs it, you can go through this process called glucogenolysis, and you can break it down, and that's helpful. And that's glycogen, okay? Then we have this thing called the Golgi apparatus. So this is where ribosomes kind of more or less come to mature. A lot of RNA products will happen here. And a lot of packaging happens at the Golgi apparatus. A lot of people call the Golgi apparatus the post office of the cell. Uh, Things go in, they get packaged up, and they get sent back out, right? So that's kind of the Golgi apparatus, okay? Intermediate filament. So these vary. This can be like stuff that holds the cell together or helps with like DNA replication. Yeah, you don't really need to know a lot about that. Microtubules, again, DNA replication, cellular structure. Microfilaments, again, DNA structure or a DNA elements with how the cells divide and stuff like that. That's those microtubules, microelements, microfilaments. Now these are used in like muscular cells too, to a larger degree, but not super high yield. Your book kind of talks about what you need to know about those. Now the rough endoplasmic reticulum has these little ribosomes, these little protein guys stuck to it. Okay, so the RER, the rough endoplasmic reticulum, has protein stuck to it. Okay, and basically that its job is to hold on to proteins and kind of do stuff with them. Okay, I don't want to get into a lot of detail on these. And then lastly, or not lastly, but the smooth endoplasmic reticulum. This is where like fat soluble things are made, specifically hormones. A lot of hormones are made on the smooth endoplasmic reticulum. So like, for example, the testicles or the adrenal glands, they produce a lot of hormones. So a testicle or an adrenal gland is going to have a lot of smooth endoplasmic reticulum compared to, I don't know, your heart, like your skin, something like that. It's not going to need a lot of hormone, for lack of better words. Okay. And again, you're going to have to give me some grace on these. I don't want to tell you a bunch of stuff you don't need to know. Cytoplasm here, we're here. Basically, this is just like the gooey stuff inside of a cell that holds everything together. It's kind of watery, kind of gooey. It depends. But that's kind of worth knowing. It holds the cell together. So I imagine it kind of jello-ish. Okay, gooey. The plasma membrane, like this is the membrane of the cell. Okay. The mitochondria, we've heard of this, is the powerhouse of the cell. This is where the electron transport chain takes place. There's a lot of other reactions that take place here, like oxidative phosphorylation takes place here. So basically, we can take a little bit of something. We're going to talk about this at the end of this lecture, and we can push it across this gradient to create heat, water, CO2, and energy, ATP. So that takes place in the mitochondria. Okay. Now keep in mind, a lot of cells have more than one mitochondria. I want to point out. Okay. Centrioles, these are weird. These are basically, again, used in DNA replication. Okay. They act as like poles to help pull the DNA apart so it can replicate more or less. Okay. A vacuole is just like a little canister, more or less. They can hold stuff, whether that's fluid or cellular machinery or whatever needs to go in there. It can hold stuff. Okay. And lysosomes, these are like the little trash men of the cell. Okay. So they go around the cell cleaning it up. They'll eat old parts of the cell that are dead or not dead, but their life is over. They need to get destroyed. So it'll go over there and clean up stuff. It'll find cellular trash and remnants and debris, and they'll just pick them up and destroy them. So that's the cell in a nutshell. Well, there's a lot of videos online that talk about specific cellular functions. I will say I think the book does a pretty good job on what you actually need to know, as opposed to like, you could watch literally a 2-hour video on just the nucleus. But I don't know, that's not worth your time, really. So keep that in mind. Now, the cell membrane, this is occasionally tested because we're going to talk about like facilitated diffusion and all those things. But basically, our cells are obviously encapsulated, okay? And there's a membrane, right? So there's like this sac around the cell membrane, and it allows certain things to come through and not, right? So it is semi-permeable, right? So to permeate is to pass through. So it's kind of permeable, semi-permeable, and it has a selective membrane, right? So it will select, for the most part, what can and cannot come through its walls, okay? And what determines whether it comes through the walls or not is its charge. So the charge of glucose or sodium or a protein can determine that. Its size of the glucose or the protein or sodium can determine if it can squeeze through there. Whether it's made mostly of water or made of fat determines if it can get through the permeable membrane. So there's a lot of ways that determine what can and cannot go through the cell. Okay, um, the book gets a little excited about this for no reason, but there's these things called hydrophilic layers. Okay, that's the outside of the cell. So it's these little heads, these little phosphate heads right here. Um, Basically, the hydro water, philic means to love. So it literally means, again, medical terminology, to love water. Okay, so to love water, part of the cell is the outer layer of the cell. Um, as opposed to the hydrophobic part of the cell, here literally means hydro, water, phobia, afraid of, layer, right? So this is the part of the cell that's afraid of water. So it literally means that it's afraid of water. And so this area right here is actually made of fat. And as you can imagine, fat and water don't mix very well. So they tend to not get along. So this influences, again, why some things can get through, some things can't, some things need a protein, some things need energy, and why all these different things happen. Excuse me, it's because of this layer mostly, okay? And also, I'll point out these things. They're probably showing you like receptors here. This is a receptor, okay? So receptors sometimes, if there happens to be the right thing floating through the blood and it's the right charge and it's supposed to, it'll bind to a receptor like this, and it might send a message through the cell, okay? Or it might send a protein through or whatever it is, a hormone. Hormones don't need these, but um, so that's kind of worth knowing. There's receptors on the outsides of cells, and you'll notice that these are different shapes because they do different things, right? So this, for example, arbitrarily might be a testosterone receptor, and this might be an epinephrine receptor, and this might be, testosterone receptor is not a good example because that's a hormone. But, this might be, I don't know, a methamphetamine receptor. I don't know. So I'm just saying there's different examples of, receptors, and they're expressed in different locations on different cells based on the job that the cell does, okay? So, again, I told you what the cytoplasm is, right? It's this fluid-like material. It's kind of gel-like inside the cell. And the cytosol is the actual fluid-like portion of the cytoplasm, okay? A little bit of a distinction there that you probably need to know. Now, organelles, these are these cellular machinery things I was just explained, right? So the lysosomes and the centrioles and the mitochondria and the nucleolus and the Golgi and all these things inside the cell that do, these are the organelles, okay? And I just explained this. So it's not unreasonable for you to know the things in the book. It's literally like a couple pages. So you should know that. Probably, especially the high school EMT students, this shouldn't be like mind-blowingly complicated because you've covered this in high school. So it's not like I'm not asking you to understand medical school level biochemistry or college level biochemistry even. It's pretty reasonable stuff. So you can knock this out. Now, there's a lot of YouTube videos out there in the world that if you want to dive deeper into this, power to you. I don't think that's the goal for today for me to give to you. So I recommend channels like Ninja Nerds is a really good channel. There's a channel called Dr. Matt and Dr. Mike. It's a good channel. Speedy Pharmacology is a good channel. So there's all these channels. Khan Academy is a good channel. That you can just type in what is a peroxisome, right? It relates to protein breakdown. What is that, right? And you could go find out what that is exactly, okay? So yeah, there's different resources out there if you want to dive deeper into this, but I'm not doing that today. So again, the nucleus, it's typically the largest structure inside the cell. Really depends on the function of the cell. It serves as the control center. Again, our DNA is the program that's telling your cell what to do, and that's stored in the nucleus. So it kind of sends out messengers through RNA of go do this, go do that, go make this protein, go make that. So that's kind of how that works. And it has this genetic code, right, that tells your body what to do, okay? So that's the cell, okay? If you want to dive deeper, like I said, there's a lot of YouTube channels out there. I just don't feel like that's what I need to do for this lecture. So feel free to dive deeper. I'm not doing it for you. Now, let me booyah over here now. So they want to talk about the life cycle of the cell. They've kept this very simple. So keep it simple, okay? So the they want you to know this whole interphase, mitosis, cytokinesis, differentiation thing. That's cool. So interphase, right, is the area prior to, or not the area, but the time prior to division, okay? Prior to going into mitosis. So interphase is when the cell's kind of growing and it's gathering the nutrients it needs to get ready to replicate, okay? You know, most cells, if they do divide, can divide like 40 to 60 times, okay? Ish, depends. But not all cells divide, okay? So like brain cells as an adult, they don't divide. Heart cells as an adult, they don't divide. Um, but like your skin cells and your hair cells and the cells in your intestines, they divide almost constantly. Okay, so they are dividing really quickly. So it just again, it depends on what the job of the cell is. So interphase again is when the cell kind of gathers the things, whatever that is, it needs to get ready to divide. Okay, and at some point, there will be a trip in the machinery to say, to pull it out of a station, you know, in its quiescent phase and say, hey, it's time to go. Like, it's time to start replicating. Why that happens, how that happens, not important. But there will be a point when cells say, time to go, time to replicate, let's do it. And then we enter this thing called mitosis. Okay? Not to be confused with meiosis. We'll talk about that. But mitosis is when we get daughter cells. Okay? So we take one Apple Pencil and it does its thing, and we end up with two more Apple Pencils. Okay? Or another Apple Pencil. Okay? It divides. Um, so that is a daughter cell, right? It's just like it's just a replicate of a cell. Okay? It should have the same DNA, should have the same machinery. It should. Okay? That's what's normal. That's physiology. Okay? So this cellular division, you know, so you go from interphase right here when the cell's kind of like, all right, I'm gathering the nutrients to get ready to divide. And then we go into prophase, metaphase, anaphase, telophase, right? I remember this by the acronym PMAT. Okay, so you go to prophase and metaphase and anaphase and telophase. Okay, and basically what's happening here is you're getting the DNA to replicate and properly go to two sides of the cell to where eventually you can have two full sets of DNA within the cell as it's getting ready to pinch off, and then cytokinesis happens. Cytokinesis literally means to split, right? So cytokinesis is when the outside of the cell, the cytoplasm, right, literally splits. It splits into two, and that is cytokinesis. Okay, so at this point, we should have two of the same cells, and then differentiation can occur later on. So differentiation, um, there's a lot. I mean, you know, you have differentiation when you have children, right? That's one way differentiation can occur. Mutations, environmental influences, right? It's like the sun or cancer gene or cancer causing agents like carcinogens. There's a lot of different ways differentiation can occur. I don't really want to go off into that, but differentiation can or cannot occur. It kind of depends. Okay. I just explained this to you. Um, but the thing that's different on here is this word called meiosis. Okay. So what I just described to you on the previous slide is mitosis. Mitosis is daughter cells, okay? Meiosis is sex cells, okay? So in men, this would be sperm. In women, this would be eggs, oocytes, right? So these are like sex cells. And um, what happens in a sex cell is you end up with like half of your DNA pushed into a new cell. So so typically there's 46 chromosomes in a cell. So when meiosis occurs, right, sex cells, like sperm, then instead of being 48 six cells in there like normal, you end up with 23 and 23 in two cells, right? So they split your DNA in two. So you end up with 23, 23. And then mom will have another egg with 23 chromosomes in it. And so it happens when mommy and daddy love each other, they make a baby, these join together, and this is how people are made. And this is why we're all unique. Because you got half of your genetic code from mom, half of you're supposed to get half your genetic code from your dad, and this comes together and this makes humans unique, right? This particular computer code or DNA is what creates the uniqueness of humans. So again, I'll recapitulate. Meiosis, sex cells, 23 chromosomes. Mitosis, daughter cells, identical cells for the most part that are created. Okay, 46 chromosomes in those. That's pretty much what I'll say about that. Now, Cell division should, as cells die, cells should be replaced, right? That's logical, right? If cells died at a really quick rate and we weren't replacing them, you would die. Um, but occasionally we can have this scary medical word, the C word, not what you think, called cancer. And cancer is also called, it can be called neoplasms, okay? Now, a neoplasm literally means new. Plasm is like talking about like growth. So it literally means like a new growth, or like a new something is probably a better way to say that. So a neoplasm is like a cancerous growth. Okay, so this could be a breast lump. This could be testicular cancer. This could be brain cancer. Right? It really kind of depends, obviously, um, on what the cancer is and what the lump is. But luckily for us, every before a cell divides, our body does a really good job of like checking it out. And we have all these pathways in our body that you don't need to know about that basically kind of scans the DNA before we go ahead and go into mitosis. That's important. And then as genetic replication is taking place, we're checking it several times for proofing, like we're checking it to make sure the code is correct. And then even after it's done, it's being checked again. And throughout the life cycle, before it can replicate again, it's being checked. And if the DNA gets jacked up from radiation or carcinogens or smoking cigarettes, don't smoke cigarettes, then it can say, all right, life's over because this particular cell's DNA has been damaged and we don't want cancer. We don't want bad cells to be reproduced by this bad cell. Because remember, a bad cell will, in turn, if it doesn't get stopped in the checkpoints, it'll produce bad cells, and that could produce cancer and pathology and all these diseases. So what happens is with a neoplasm, you essentially have a cell that is a bad cell that replicates, and sometimes that replication can go unchecked. And when that replication goes unchecked, you can end up with malignancy, right? And malignancy is a bad cell that is replicating quickly, okay? So malignant breast cancer, right? But we can also have what's called a benign neoplasm. So a lipoma, right? A chunk of fat on someone, or a mole, right? So like a skin freckle, right? That is a benign neoplasm, technically, usually, unless it's like melanoma or basal or squamous cell carcinoma. But basically, a mole, like a normal freckle, for example, that's a lot of melanin that's replicating, and it really shouldn't. But it's kind of caught before it can get out of control. So your body's machinery is able to say that's enough, and it stops. And then when it does stop, you just end up with a benign mole, right? So that's kind of an example of a benign something. We have benign breast cancers, right? So there's just a lump of fat or mass in someone's breast, and it's benign. We have benign endometrial cancers, like a leiomyoma, right? There's all these different benign things that exist in medicine, and sometimes they can become malignant, right? And so benign things can become malignant. Malignant things typically don't become benign, but the distinction is worth knowing. And the word that confuses students is the word metastasis. So metastasis is when you end up with like, we'll just give an example of colon cancer, right? Let's say you have colon cancer, okay? And the colon cancer should be in your colon, but since your colon has a rich blood supply, that cancer can get sucked up into the blood and travel to the liver and say, good place to park it. So your adenocarcinoma of your colon can pick up, travel through the blood, and go to your liver, for example, or your lungs, or your brain, or wherever. And it can park it there and say, all right, I'm going to start creating cancerous masses here. That's an example of metastasis. Metastasis is when cancer picks itself up, usually through the blood or the lymphatics, usually, and it will pick itself up and move to another part of the body. That's metastasis, okay? That's enough on oncology, but that's an example, okay? Okay, cellular respiration. This is what EMT nightmares are made of, okay? It's very scary, it's very frustrating, but we're going to get through this because we literally only need to know the bare basics of this, okay? So let's think about this. Cells respirate. Oh, yeah, that's why we have to breathe, right? So cells respirate. Respiration takes place at the cellular level. Ventilation takes place in and out of the lungs. There's a difference. But cellular respiration is the process of delivering of creating energy more or less at the cellular level, okay? And the ingredients to this delicious recipe is glucose, one, oxygen, two, and kind of water. Um, and so you need these, really, I'll just say glucose and oxygen are really what you need um, for cells to do their job. Because in order for a cell to do its job, it typically has to have energy. And as we know from ninth grade biology, energy in the body is stored in the form mostly as ATP, right? Adenosine triphosphate. So in order to create ATP, adenosine triphosphate, we have to give the body glucose and oxygen. Some magic happens, also known as cellular respiration, and we can turn that into energy for cells to do their job. Now, the first piece of this that the book really kind of neglects is to eat. So you can eat a banana or fruits or a candy bar or whatever. At this point, we're just focusing on glucose and get glucose dietarily, right? So you can eat and you can end up intaking some glucose, and that comes in through your body, and eventually this will, through a long process, end up in your blood. You end up with glucose, simple glucose, right? Very simple sugar in your blood. And through a step of reactions, you can take this glucose and you can break it into stuff, which I'm going to show you in a couple slides, that can then go down into the cell and produce a lot of energy if the right ingredients are there to make this cake. And so that's kind of the beginning of this. So I'm going to talk about this, okay? But let me just show you the next slide. So this is all you got to know, right? Just memorize this by tomorrow morning and you should be fine. No, I'm just kidding. Do not memorize this, okay? Do not memorize this. This is a joke. But I want to illustrate a point here. Your body doesn't just run on glucose, right? Like your diet's not just glucose, is it? Right? It's glucose and protein, and carbohydrates and fat and all these other things. But in medicine, we tend to focus on glucose because it's a really rich source of ATP for our body, okay? I want you to know that glucose can be stored as glycogen, right? So I told you about that, right? It's glucose stored on like a chain that branches to where when we need glucose and we don't have any from our diet, our body can break down this storage called glycogen, okay? And the process of that is called glycogenolysis, right? Where you break down glycogen. But sometimes we don't have glucose because we haven't eaten today, and sometimes we've burnt through our glycogen because we didn't eat yesterday or the day before. And sometimes your body will say, I'm hungry, I need energy. So it can break down fat, okay? And when we break down fat, ketolysis, right? Like the keto diet. Oh yeah, the keto diet breaks down fat. You can end up getting energy from like glycerol and fatty acid and stuff like that. We don't even talk about that is. And that's one way it can eventually shun its way to the same process I'm about to talk about, right? The TCA cycle. And you know, there's we can use straight up fat for energy sometimes. We can use protein for energy. We can use glucose for energy. So we can use all these different ways to create energy. Well, the consequence of all of these is they create byproducts. So sometimes we have to take these byproducts and through the urea cycle or through these different pathways, like the fructose metabolism pathway, or you know, all these lactose and lactulose and all these different pathways. They all are connected in weird ways that you don't need to know about. But I want you to be aware. The book focuses on glucose, and that's fine because we actually see this a lot pre-hospital. People get low blood sugar, high blood sugar. But I want you to know that we get energy through a lot of different ways, okay? And what this picture on the slide is trying to show you is how connected these pathways are, okay? Sometimes we make glycogen, sometimes we use glycogen, sometimes we use glucose, sometimes we have to take, you know, from this cycle to still to this cycle to make energy for that cycle. They're all connected, okay? And interestingly, pathology, right? Bad things, diseases, can happen in almost any part of this, right? If you're missing just one of these enzymes, you're messed up. You're sick. You have some type of metabolic disorder, okay? Or if you have too much of this one beta hydroxybutyrate, right? It's a ketone. You get ketosis, right? You get diabetic ketoacidosis, right? So what I'm saying is they're all related, and I just want to point that out. That's what the slide's about. Again, I'm kidding. Don't memorize this. You really do not need to memorize this. But you do need to know this, okay? So let me move my little camera back over here. So this slide, I think it's okay. It's pretty fair game. Okay, so you eat your glucose. Okay, glucose comes into the cytoplasm of the cell. Okay, how is that done? It's done through insulin. Allows glucose to enter through that selectively permeable membrane through facilitated transport, facilitated diffusion, to enter, and we're going to talk about that on the next slide, to let it go through into the cell. So we get glucose in the cell. Okay, we're at that part. And what happens is, without oxygen, so this is anaerobic, okay? Anaerobic meaning ana means without, aerobic means with oxygen, okay? So it's literally without oxygen, anaerobic, as opposed to aerobic is with oxygen, okay? So you got to know that distinction. I'll say that again. Ana means without. Anaerobic means without oxygen. Write that down. Get a tattoo of it. You got to know that. Anaerobic means without oxygen. Aerobic means with oxygen, okay? Now, what happens is glucose enters the cell into the cytoplasm. That's fine. And there's this thing that takes place. It's kind of complicated, called glycolysis. Let's break down that word, glycolysis. Glyco, glucose, which is a sugar. Lysis. Lysis means to split or to cut. So we're kind of chunking up some glucose, okay? And the byproduct of this reaction is something called pyruvic acid, okay? And in the process, in this anaerobic process that's taking place in the cytoplasm called glycolysis, we have a little bit of something that's made. We have ATP. That's really good. That's a really good currency for energy. And we also get this thing called NADH. Okay, we get two of those. And I'm going to come back to what we do with those in a minute. But I'm going to keep going this way. So after the anaerobic process of glycolysis, since it doesn't need oxygen yet, we get pyruvic acid. Okay, now pyruvic acid is this really great structure that can go a lot of different ways. And one way that it can go is it can go south, right? It can turn into the mitochondria and become acetyl-CoA. That's the word, acetyl-CoA right here. Excuse me. It can become acetyl-CoA. And acetyl-CoA can turn into this stuff that you don't need to know about and enter this thing called the citric acid cycle or the Krebs cycle or the TCA cycle. Yes, all three names, annoying. The citric acid cycle is the same thing as the Krebs cycle is the same thing as the TCA cycle. I'm not the one that came up with this, okay? So don't take it out on me. But they're all the same thing. You got to know that. They're interchangeable. And through this process, we can go from pyruvic acid to acetyl-CoA through the citric acid cycle, and it kind of poops out some stuff. But one of the things it poops out is CO2, and it also poops out some ATP. Again, ATP, adenosine triphosphate, that stuff that gives us energy, okay? And that's cool. That's great, okay? But this process does require oxygen. In order for the TCA cycle, citric acid cycle, Krebs cycle to take place, it requires oxygen. So I'll recapitulate. Pyruvic acid goes into the mitochondria. At this point, there's some oxygen there. Turns into acetyl-CoA. CO2 is produced. ATP is produced. A little bit of water produced. A few other things are produced, but that's not important. And that's basically what happens. So we started with glucose. Glycolysis happened, pyruvic acid, acetyl-CoA, and then citric acid. That's kind of what I want you to know. This isn't the presence of oxygen, though. It is aerobic catabolism, right? With oxygen, okay? Now, if you don't have oxygen present, or the oxygen is not enough to meet your demands, you can shift pyruvic acid to lactic acid. And this does produce a little bit of energy. How much? Not important. So, have you ever went sprinting before and the next day you're super sore? Well, what happened was when you were sprinting, your muscles were using more, they were requiring more oxygen than what your little heart could give them. So really quickly, your body said, we don't have enough oxygen. Don't send it to the Krebs cycle. Just shun it over to lactic acid. Or you end up with lactic acid, basically. Through anaerobic metabolism, because we need energy now, we don't have what we need. And that lactic acid is sitting on your muscles and it causes pain. So imagine acid on a muscle. Of course, that would be painful. So that's how you end up with lactic acidosis. So sprinting or lifting weights would be one example. of how you could get lactic acidosis. Another way you can get lactic acidosis is not having enough oxygen, right? So hypoxia. Shock, right? Shock is defined as the lack of perfusion to cells. So we'll cover that when we get into the shock pathophysiology, but I'm laying a foundation now, okay? So it shows up. Now, um, lactic acid can go back to pyruvic acid, and they kind of shift back and forth, but that's one way that this happens. So far, you might be thinking, well, through glycolysis, we get 2 ATP. Through the citric acid Krebs cycle, TCA cycle, all the same thing. We get 2 ATP. That doesn't seem like a lot. Really, it's not. Because the big boys are made in the electron transport chain, right? So within the mitochondria, the powerhouse of the cell, we have within this membrane, it's called the inner membrane right here, we have this thing called the electron transport chain. And as you can imagine, electrons, right, their charges, can produce energy. Okay, just keep that in mind. And so what happens is this NADH, through glycolysis, which I told you glycolysis is anaerobic, can shunt its way into the mitochondria to the electron transport chain because it knows if I can get to the electron transport chain successfully with oxygen, I'm going to make a shitload of ATP, okay? It can make 32 ATP to be exact. And that's really useful for your body. So what happens is this NADH, which is like a currency of energy, right? Goes through this really weird process you don't need to know. And basically there's this gradient that takes place with hydrogen and electrons, and basically energy is produced. And heat and water and I think, no, not CO2. Maybe CO2 is produced there. I think CO2 is produced there. Don't get me lying on that one. But it's been a minute since I studied the basic sciences. So what's important is water, heat, energy. Those are the big things for an electric transport chain. So what happens is you end up with 32 ATP in the presence of oxygen when they can meet up at the electron transport chain within the mitochondria. Okay, oxygen has to be present for that to take place. If oxygen's not there, this doesn't happen. So you ask yourself, well, what if someone doesn't have oxygen? I'm glad you asked. They die, right? This is why people die when you don't have oxygen, because your body can produce a little bit of oxygen or ATP without oxygen, but it's not enough. So that's why, obviously, you need oxygen to live. So I have here a little bit of a note that I put in here, right? Your book and what I'm saying, there's a little bit of discrepancy with what I'm telling you, but understand this. This process of the electron transport chain and the citric acid cycle and glycolysis isn't free. It uses a little bit of energy for these things to happen. They produce more energy than they use, but there's this thing called the net, right? So you end up netting 32 ATP ish when this thing's over. If you do anaerobic ATP, so for one glucose, you can get up to 32 ATP if there's oxygen present, aerobic metabolism, okay? That's a lot. In comparison to if you don't have that, if you don't have, if this whole bottom area doesn't exist because it requires oxygen, aerobic metabolism, if you don't have that, you only end up with 2 ATP, and that's not near enough to sustain life. So that's why this happens, okay? Kind of explains that. Now, I want to point out some things. Your brain uses a lot more ATP than your hair, okay? So this is where I'm telling you, cells are different, and certain cells will have different requirements and needs and demands and blah, 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 blah. So just be aware of that, okay? Now, I explained all of this to you. I don't want to read it to you again. Yeah, I talked about all that. Now, we have, so that's cellular respiration, okay? Cellular respiration takes place at the cellular level. Ventilation is air in and air out of the lungs and the mouth and the nose. Now, there's this, hang with me, we're almost done. Cellular transport mechanisms called, like there's, endocytosis and facilitated diffusion, all these things. I'm going to talk about those, okay? But basically, we got to talk about how does stuff, proteins, hormones, glucose, stuff, get through the cell and out of the cell. There's a process that you kind of need to know because we can influence this in medicine. So our cells have this permeable membrane, selectively permeable membrane, again, where we let some stuff in, but we don't let other stuff out, and blah, 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 blah. So concentrations tend to influence this, charges tend to influence this, sizes of the molecules tend to influence this, stuff influences this, okay? Receptors, enzymes, a lot of stuff influences this. But the first one we're going to talk about, one of my favorite examples, is diffusion. So we've all been there. When the nasty guy in the locker room or in the classroom or wherever sprays their nasty, disgusting Axe body spray all over their nasty body, and it's at an area of really high concentration around them, like it stinks. It's really concentrated around this guy. We'll call him Ken. And so what happens is you're over here minding your business in the other corner of the classroom, and over a period of time, the area of high concentration of Axe nasty body spray is going to make its way to an area of low concentration to your innocent little nose on the other side of the classroom, and you're going to end up having to smell it. Okay? That's diffusion. Diffusion is when that area of high concentration of Axe body spray molecules in the air made its way to an area of lower concentration to your innocent nose. And so you had to smell it. Same could be said with farts. So that's the example of diffusion. But the example of how this happens in the body is with oxygen and carbon dioxide. So in our blood, in our lungs, cell or blood is bringing back CO2 because that's the byproduct of cellular respiration. So in our blood, on the venous side, coming back from the cells to the lungs and the heart, we have a high area of CO2. And in the air that we're breathing in, there's a high area of oxygen. And so through simple diffusion, it's going to go from areas of high concentration to low concentration. And when this happens, that's what's happening in the lungs. That's what we're trying to get to happen. So this happens a lot throughout the body. And simple diffusion example is CO2 and oxygen in the lungs. Simple diffusion would be the nasty guy with his Axe body spray or ripping farts in the classroom, and your nose gets destroyed by it. That's what's happening. It's moving from an area of high concentration to an area of low concentration. Okay? Now, filtration, we're going to talk more about diffusion, but hold on. Filtration is simply filtering something. I don't think I need to explain this. Have you ever made coffee before? That's a filter. Now, what let me just go here real quick, and I'm going to go back. I should have fixed this slide. Forgive me. We have these things called facilitated diffusion. Let me see where we're at. Yeah. This is what I want to talk about, and then we're going to go back. We have this thing called facilitated diffusion. I'll just explain this instead of reading it to you. And where this happens with facilitated diffusion, I'll write this down for you. A good way to think about this is insulin. So you've probably heard of the hormone insulin. It's produced by your pancreas. And what happens with insulin is, so glucose cannot just go into any cell at once. So to go into like your liver cell, your muscle cell, you have to have insulin. And insulin is kind of like a key that opens up the door and lets glucose go in. And so without insulin, glucose just stays in the blood. It cannot enter the cell. So the only way it can enter the cell is if insulin's there to open up the door and let it go in. And you're seeing that here with facilitated diffusion. We're taking an area of high concentration. In this case, the blood has an area of higher concentration of glucose. Insulin is, in this particular case, facilitating the diffusion. It's saying, I'll open up the cell so that glucose can go through from an area of high concentration in the blood to an area of low concentration in this lonely little cell. That's facilitated diffusion. That's a good example of it. Now, there's another form of diffusion where we're going from an area of high concentration to low concentration diffusion. And this is called active transport. And one way that they're giving an example of this, I don't want to use this example actually. I want to use my own. So we have these things called, actually I will use this example and I'll give you an example here in a minute. They have an example of using iodine. So with active transport, you're typically transporting something against a gradient, or you're transporting something that's charged, it's having to pass through something that doesn't really like charges. You know, there's some type of circumstance going on to where you're going to need a little bit of oomph, a little bit of push, a little bit of energy to get this thing through an air through the membrane. Okay, and in this case, we tend to use ATP to do that, right? ATP is our currency of energy. Okay, it's the dollars and nickels and dimes of our body of energy. And so what happens in active transport is we take energy to allow, in this case, iodine, which happens to be charged, through the outside of the cell and get it inside a cell. Well, again, there's a charge here, so what we have to do is we have to sacrifice some energy, ATP, to allow that to happen. So we have to actively, with energy, ATP, transport it through the cell. And that's how and why that happens. That's active transport. So the other way that this happens, active transport, would be like the sodium potassium pump. Okay, so like we have these pumps in our body that pump stuff in and out of cells all day, and that requires energy. And fun fact: when we start to die, right? So like chest pain or your brain starting to die, swelling occurs because that pump quits working because it requires a lot of ATP. So that's one reason we get swelling of like tissues as they die, because the ATP isn't there. So the sodium potassium pump requires ATP. It doesn't have ATP, so things start to swell because this pump's backing up, and that's one of the hallmarks of pathology, which we'll talk about in the next chapter. But lastly, the book, I mean, they talked about this thing called um, uh, endocytosis. Okay, endocytosis has this umbrella term, and it kind of means like to take in. Endocytosis, we could say, to take in. It happens in mainly three different ways, and I want to talk about these. The first one right here is called phagocytosis. Okay, some places will call it phagocytosis. I don't care. Phagocytosis, pick your poison. And basically, phagocytosis takes place where you're ingesting a solid particle. We actually do this, white blood cells do this with like viruses and mainly bacteria, to where we'll just go and Pac-Man that little mother and just go eat it up. That's phagocytosis, okay? We do this for certain things too, but bacteria is a really good example with white blood cells. You know, white blood cells are constantly going around looking for stuff to eat that's bad. One of the ways we do that is through phagocytosis. Penocytosis means to like pinch or to drink. So you might think, you know, you kind of pinch and drink your drink. I don't know. But that means it's fluid. Okay. Penocytosis is more fluid like, and it forms what's called a vesicle. Whoops. Here. Okay. And then this last one is endocytosis. Okay. Now, this is an example of receptor mediated endocytosis. So basically, if there's receptors and the stars align, they can basically line up on here and be like, oh, my receptor fits to your receptor, let's do it. And they will close in and engulf whatever that is. So that's different examples of endocytosis. I wouldn't say these are hugely high yield, but they're in your book, so here I am talking about them. Now, moving forward to osmosis. Osmosis is, so you have to leave diffusion behind because now we're changing gears. We're going to osmosis, okay? So let me read this to you. Osmosis is the passive movement of a solvent from an area of low to high solute concentration. Okay, and we use this a lot in our body for like IV fluids, and we see this in a lot of pathologies. Okay, so that's why it's covered. So let me, I'm trying to think of the best way to explain this. Okay, so let's just do it. So these little red things on your screen, those are your little salt ions. They're little particles. And this is a bag. You know, you can imagine it's like a condom or something, just a bag. And what happens is this particular bag will let water through, but it won't let salt through. Okay? And our cells in our body tend to behave this way. Our body will tend to let water through pretty easily, just straight water, but it tends to not ever let other things through super easily. Okay, for the most part. And so what happens is these almost want to like balance out in a weird way. They almost want to balance out. So with osmosis, you end up with water moving to try to balance out like concentrations. Okay, so what you're doing is you're moving where you have a high amount of water to an area with less water to try to balance out the water concentrations, not the salt concentrations so much, but the water concentrations. So osmosis, I want you to think more, we're talking water. When I was talking about diffusion, I was talking about particles like Axe body spray and feces and farts, but we're not talking about that with this. We're talking in osmosis, we're talking about water. So let's just see why this is relevant in modern medicine and as an EMT. So I'll have you know, I'll just give you some examples here. Oops. All right. Our normal cell is made up of about 0.9% um, sodium chloride, right? NaCl, right? Sodium chloride. So when you go to the doctor's office or when you ride on an ambulance, all the IV fluid on the ambulance is 0.9%. Okay, so that's called a word isotonic. Okay, whoops, isotonic, yeah. So it's isotonic, right? So I can inject you with normal saline, 0.9 normal saline, and you're not going to have a shift of water in or outside of your cells because what I'm giving you is matching pretty much what's inside your cells as far as tonicity, right? Like the amount and tonicity, right? The concentration of a solution or its ability to draw or give water. So what we typically give on an ambulance is very similar to what is given on. This is my little background. I just moved. Um, Y'all can't see in my house. Um, Whoops. So it's pretty similar to what's given inside uh, your cells, what we carry on an ambulance. But that's not enough. Examiners don't care about that. They want to make it complicated. So what we have to know is what happens, in this case, the model is a red blood cell. If we give an IV fluid with a lot of tonicity, so a lot of salt. And what would also happen if we gave something with a small amount of salt, less than what's normal in the blood? So I'll draw this out for you. It's easy. It's really easy if you just take a deep breath. It's gonna be okay. So this oh, it's right here, isotonic. I didn't even see that. If I were to give a patient, let's just call it 3%. Can we agree that 3% is more than 0.9%? The percent is talking about how much salt is in there. So we're giving a lot more salt, about 3 times more salt in this IV fluid I'm about to give a patient. If you give a hypertonic solution, right? What's going to happen is these red blood cells are going to say, oh, there's a lot of salt outside of the cell, so I can try to balance out this water situation by giving my water outside of my red blood cell to try to balance the amount of water outside because there's more salt in that water than there is water. Okay, so what happens is water can move out of the red blood cells, and the red blood cell consequently will shrink. Okay, so water's when you give something that's really salty, water will move out of the cell to try to go make this salty water not so salty. Okay, because water can move, salt cannot in this case. So that's an example. So what happens is the cell gets really small. Now, I'll move over here. Now that so that's one way this happens, right? And that's a hypertonic solution will cause the cell to shrink. Okay. Now the opposite could be said with if you were to give like a hypotonic solution. So if you were to give like 0.45% saline, you know what's going to happen? Your cells are going to say, "Oh, there's more salt in the cell and relatively less water compared to outside of the cell." So what happens is the water that you just injected into the patient will move into the cell. And when that happens, the cell can swell. And it can swell so much, in fact, it can lyse or it can burst, right? So that is an example of hypertonic versus hypotonic solutions and what happens to cells. There's a cajillion, million, bajillion, trillion different videos on YouTube on explaining all of this. Excuse me, I'm thirsty. And so if you want, go do it. Go watch some videos on that. But that's what I just told you is a pretty adequate explanation of what you actually need to know. This is the last slide. Now, there's this balance that takes place in our body, of body fluids. And for whatever reason, the National Registry and your textbook manufacturers and the Q bank makers are obsessed with these weird factoids, okay? So this is very heavily tested for whatever reason. I don't know why it is, okay? So they like to ask, what percentage of the total body weight is made up of fluid? You know, and it's like, if we're intracellular fluid, you know, and it's just, so they'll ask weird questions like that. It's not me writing these. I'm just letting you know. They like to ask those. So I'll just read this to you and we'll talk about it because it's kind of logical. If you apply intuition, it's logical. So intracellular fluid. So let's break down what that means. Intra means within. So within the cell fluid, oh, so the fluid within the cell, right? Um, is around 40 to 50% of your total body weight. So like I'm 170 pounds. Shout out me. So if you divide my weight in two, what is that? That's like I don't know, 85 pounds. So 85 pounds of me is made of water. Okay. Um, so or I'm sorry, it's made of water that sits inside my cells. Inside my cells, about half of my weight is sitting of half of my weight is water that is sitting inside my cells. Um, and so that makes up 75% of your total body water. Okay, so I hope that makes sense. I'll say it again. Half of your weight, if your total weight, let's say you weigh 200 pounds, about 100 of that pounds is water inside cells, intracellular fluid. Now, of that water, um, if you have 100 pounds of water, 75% is your total body water. Okay? The slides may make more sense than me saying this because I'm going to be saying some very similar words. This is copied straight out of your book. I just added this from your book because I've seen this tested. Now, the extracellular fluid, so think about that. If someone's being extra, they're kind of out there. So it's outside of the cell fluid, okay, makes up 25% of your body fluid. So this is like, this has two different ways that this happens. Around 4.5% of your total body weight. So again, let's just say you weigh 200 pounds. 9 pounds of you is made up of this, your extravascular or your intravascular fluid. So intra again means within, vascular, your vessels, fluid. So your plasma. So around 4.5% of your total body weight is made up of fluid. And that fluid is called plasma. And lastly, you have interstitial fluid. So interstitial means within the middle of the cells. So if these are all cells, in between the cells, there's actually fluid in between them. Okay, that's interstitial. And that's normal. Sometimes in like heart failure and stuff, it's not normal. But for the most part, we should have interstitial fluid. And that's around 10.5% of your total body weight. So again, I hope I didn't just confuse you by saying all this. If you're screenshot this and you read it a couple times, it's a lot more, probably makes more sense than what I just said. But we have this like homeostasis that takes place with how we maintain where we keep fluids and what's the tonicity of them and what's the concentration and the thises and that's of the worlds. So we have to maintain homeostasis, and we can do this through several ways, right? We can do this through thirst and the kidneys and like water shifts, but the one that there's pathology most related to is antidiuretic hormone. This thing right here. Let me just show you it right here. Okay. So let's break down what this word means. Anti means against, right? So it's not, okay? So to diurese means to pee. I'll say it again. To diurese means to pee. So if you break down what antidiuretic hormone means, it literally means the don't pee hormone, right? It's prohibiting you to pee hormone. So the don't pee hormone is excreted in weird ways. So I'm going to tell you how this don't pee hormone, also known as ADH, works. So as the salt concentration of our body increases, we have these osmotic receptors in the hypothalamus. It's an area up in the brain, specifically right around here. And basically they're sampling and it'll say, all right, brain, we need to do something. It's getting a little salty in here. It's not salty enough, whatever it is. So they can influence antidiuretic hormone. So if it's not salty or too salty, it'll tell the brain to release antidiuretic hormone or not, and then it releases into the bloodstream. And what happens is eventually this antidiuretic hormone will go down to the kidneys and say, okay, kidney, I want you to hold on to more water, but get rid of salt. Okay? And that's one way that we influence our blood pressure, and one way that we influence the amount of just general water that we have in our body is through the antidiuretic hormone, also known as the don't pee hormone. And so there's pathologies where you have too much antidiuretic hormone or not enough antidiuretic hormone, whether it's diabetes insipidus or SIDH or nephrogenic diabetes insipidus. There's all these different pathologies, and that's why ADH tends to get focused on because it does also influence blood pressure really largely. But that's kind of what I think you should know. I mean, we could talk about this a lot. There's a lot of, like I said, there's a lot of pathologies related to this. So that's the next slide. I know this is a lot. Let me end with this. This isn't like wildly high yieldly tested on the National Registry, but this is foundational knowledge. Okay, so this is the knowledge that you gotta have to where when I do tell you about diabetes insipidus or syndrome of inappropriate antidiuretic hormone, right? You have to understand what ADH is to be able to like pick up like, oh, that's what he's talking about. And you're going to see this when we start getting into like gross anatomy, right? When I tell you the difference between a scaphoid and scapula and the maxilla and mandible, right? Like there's a reason we have to know these things. Um, Now on your in-class quizzes and in-class tests, I might just straight up ask you, what does ADH do? On the National Registry, they're probably not. Um, so just realize that like nothing on here that I told you isn't testable. Anatomy is very, very testable. It's super easy to write test questions is what I'm saying. So for this material. So take a deep breath. It's going to be okay. I know this can be overwhelming. This actually, believe it or not, is my least favorite part of medical school is learning biochemistry and what we just talked about. Um, Although I find it interesting, I'm not very good at it because I'm more of like a gross anatomy, gross pathology type guy. So, anyways, with that, I appreciate y'all paying attention, and we'll just keep this series going. I think it'll be like 4 or 5 videos, so we can make it into bite-sized pieces. So that's it. Thanks for